these four, some of these are I have given here. So we can how we can overcome this problem of repetitive DNA. So we can have a higher coverage in our sequencing technology. By sequencing deeper, we get more reads which will span to the flanking unique regions that will be present uh, close to the repetitive regions, and the assembler could have more information, so it can arrange them correctly. So we can sequence the genomes at a higher coverage to get out of this problem. Then we can have longer reads. Now we have technologies like PEG Bio and ONT Oxford Nanopore technologies, which can sequence longer reads around of uh, 10 kbs and 100 kbs, which will like which will span over your repetitive region. So it can sequence an entire repetitive region, and the problem of this repeat sequence will not be there. And again, we can have reference guidance. If you have a closely related reference genome to your sample, you can use that reference to guide your assembly to make a to like identify the potential misassemblies, or you can correct them. Then we can have assembly validation. So there are some tools which assess your or evaluate your assembly once it is assembled. Once your data is assembled, you can check through those tools. They will tell you that there is this percentage of misassembly in your assembled sequence. So you can use those tools also. So the repeat problem is why the assembly is still an active area of research, even after decades of work in this field. Like, this is a very big problem for sequencing. Okay, next we have is, so how do we assemble the sequences, these raw FASTQ sequences? Is so we have two fundamental different approaches to assemble the genome. So it's crucial to understand when to use which one. So let us start with the de novo one. So de novo assembly is basically you are building something from scratch, something very newly, from very beginning of something. So you don't have any reference for that. You don't know what will come out of it. So when you choose de novo, you have some advantages, like uh, you don't need a reference genome. You don't need any reference, you can directly take your sequences and you'll assemble them and you'll get your assembly. Then second is you can discover novel sequences. You will find something new if you use this approach, because it doesn't include any reference guidance. Then you can identify some structural variants in your organism. Then you can capture some organism specific genomic features. But we also have some, it also has some disadvantages, like it is very intensive computationally. It will take hours to days to compute, and this algorithm, the de novo approach, also struggles significantly with repetitive regions because it doesn't have any reference. So it has to tackle the repetitive repeat problem, and it requires uh, higher coverage in comparison to the reference based assembly. And the assembly which you will get will be sort of more fragmented. It's not necessary that you will get a fragmented assembly every time, but in comparison to reference based, it will be a fragmented, a more fragmented one. Okay, so next approach is reference based, that is guided reconstruction. So this alternative is where you align your sequencing reads to an existing reference genome from a closely related organism. So you will take your reads and you use an algorithm to align your reads to that to a particular reference which is very close to your sample organism. Then once you align, and you will find a consensus sequence, and that will be your assembled genome. Now it has some advantages that it's fast. It will take minutes to hours in comparison to de novo approach, and it works well with lower coverage, typically 10x to 30x. Then it leverages the existing genomic knowledge that the reference sequence has, and you might yeah, it's computationally straightforward, so it's bit of a simple task for the computer also. Now the disadvantages are it introduces a reference bias. So uh, you tend to find what's already in the reference. So you might miss some novel sequences, and then you also need a closely related reference genome to be there for this to equip this approach, and you might miss some structural variants also. uh, okay, so when to use which, when do you use de novo, and when you will use reference? So it is clear from the advantages and disadvantages. If you have a reference genome available, you can use reference based. But if you want something to if you identify something novel, something new out of your sequence data, so you will always go for the de novo assembly approach. Now this is the these are the visual representations of how the reference based assembly and de novo assembly takes place. So you can see in the left figure, the first section, it's showing the reference genome. I'm sorry about the blur images. It is visible. Once they are aligned, you can get a consensus of this, and you will generate your new genome. But in de novo assembly, you can see here there is no reference. So it is the algorithm will just find the overlapping sequences. You can see here these sequences are overlapping to each other, and these overlapping sequences will be used to form a consensus sequence, and that would be your uh, new assembly. So as I said, in de novo approach, you will find a more fragmented assembly. So you will get not a proper genome, the whole thing, but you will get some contigs out of it. Contigs are contiguous sequences uh, which are longer than your reads. So you can see config 1, config 2, config 3. So this is a fragmented assembly. You can find many sequences which are fragmented on the NCBI database also. Those assemblies are fragmented. So yeah. And on the right side, I have given a picture to understand it better, how de novo assembly takes place. So these are your reads, and you can see the overlapping sequences here. CG, and CG are overlapping, and here ATCG is overlapping. So it will be merged to a single consensus genome. Okay. So the difference between the reference based and de novo assembly is clear, I think. Can I get some replies or any doubt up until now? You can ask. Or the first reference.
Yes, sir.